Good to go? Okay, we are very lucky, sorry. We are very lucky to have Monika Richardson join us here again. In January of this year, she gave us the first report. Tonight, she'll discuss the results of her master's project to compare the ectomycorrhizal fungal communities of 3 Pinus species on the California North Coast. Monika is a grad student at Cal Poly Humboldt and recipient of the Dr. Harry Tiers Scholarship awarded by the MSSF. She is looking forward to finishing her work this spring and moving on to a teaching position, maybe in the Humboldt area. Please welcome Monika.
Okay. Oh. Maybe, yeah, maybe a little too dark, maybe a little more light. Yeah. Okay. Thank you guys so much for your patience. And thank you so much to the Mycological Society for having me back. Thank you to my friends and my family for coming, and for everybody else for coming. And I'm super excited to talk about this because it has taken up the last 2.5 years of my little life. And I'm ready to be done. So I hope that this is satisfying for me. I think it will be, and I hope it's satisfying for you guys too. But I'm going to pretty much give an overview of how I've done this research, what I've found, and kind of just talk about this really interesting study system that I've been working in, which is primarily Bishop Pine, which you can see here, up on the far northern coast of California in Humboldt, where I have been living. So let us begin. Some of us were at the last presentation, but I think we've forgotten by now. So let's just start with basics. What is ectomycorrhizal symbiosis and what are ectomycorrhizas? If we break down mycorrhiza, supposed to just have one arm. Myco means fungi, rhiza means root. So we're talking about a symbiosis between fungi and plants. And it's actually not just ectomycorrhizal symbiosis where this occurs, but there are a bunch of different types of ways that fungi and plants interact. And in fact, arbuscular or endomycorrhizal symbiosis is what 80% of plants on Earth engage in. And it's a more ephemeral relationship where the fungi involved are microfungi and glomeromycotina. And they are really predominant in like grasslands or tropical hardwoods. And these relationships don't last as long. They can be just a few weeks where the fungi has made this association with the root, dies off, and then a new fungi comes in, happening in multitude many, many times. But what I'm talking about today is ectomycorrhizal. And it's called ecto because the mycelium or the hyphae of the fungi doesn't penetrate into the cells of the plant. And instead, it goes around the epidermal and cortical cells and it forms this structure called a Harding net. And that is where nutrients and water are being transferred between the two different kingdoms. And ectomycorrhizas, what pictures that I took over here, are macroscopic, morphologically distinct, what I'm going to just call shared organ right off the bat. Because what this is the refined root tip of the tree that's wrapped in the mycelium of the fungus to create this setting where nutrient transfer can occur. And this is critical because what is happening is the fungi are mining for nutrients in the soil, predominantly sources of nitrogen and phosphorus, and they're transferring, along with water, they're transferring that to their plant host, and the plant, or the tree predominantly, that's doing photosynthesis, is returning carbon in the form of carbohydrates or photosynthates to the fungi. And so it's this really mutualistic relationship that's on a spectrum of mutualism to sometimes even parasitism, depending on what's going on in the ecosystem. But the fungi that do this are only in what we call the higher fungi or dikaryon. And this is an Ascomycota and Basidiomycota. Above this is a phylogenetic tree of life of fungi. Above these two big phylums are smaller microfungi. So ectomycorrhizal symbiosis, which is actually a more relatively newer form of symbiosis between fungi and plants, evolved in groups of fungi that are larger, that are more robust, that create networks that can last for longer. And our earliest fossil of them is about 500 or 50 million years ago, but it's projected that Ectomycorrhizal association probably started around 150 million years ago. But that is so much younger than the endomycorrhizal association I was talking about earlier, which is actually likely what allowed water plants to come onto land when they did, like 500 million years ago. So ectomycorrhizal symbiosis is almost, in my opinion, kind of like a more nuanced, maybe more evolved form of symbiosis that takes place with those higher phylums of fungi. What's going on here is actually critical to ecosystems, because this is a really interesting image from Hawkins et al. 2023 that kind of talks about how not only are nutrients being transferred between fungi and plants, but carbon is being sequestered. Because like I mentioned, the carbon from the tree is going into the mycelium of the fungi. And when it's doing that, the fungi are taking that carbon and it's going towards mycelial growth, but it's also being traded with bacteria in the soil. And it's basically being sequestered in this way. And it's sequestered at a much higher rate, gigatons of carbon, than the arbuscular mycorrhizal, or the endomycorrhizal association I was talking about, or ericoid, which is just associations with plants in Ericaceae, like madrone or manzanita. And so it's a fundamentally super important symbiosis, not just because it sustains the plants that engage in it, but also because it is monumentous in sequestering carbon, right? A tree takes in carbon through photosynthesis, but it's actually stored in the soil because of its associations with fungi. And so this is ectomycorrhizal symbiosis isn't done by the majority of plant species. Only around 6,000 plant species, which is about 3%, are able to engage in this. But you'll see, this is a proportional map from Kabir P's lab down in Stanford, about where these trees, or where these plants that do ectomycorrhizal symbiosis are concentrated. And you'll notice that they're mostly, besides a few remnant populations in more tropical regions, they're mostly in high latitude temperate regions where the symbiosis is really beneficial, because those are soils that are super nutrient limited. So what's happening here is, this is not a hospitable place to live, but the evolution of ectomycorrhizal symbiosis has been so successful that it has allowed for the proliferation of these plants in these areas. And you'll notice that it's pretty red where we are in Tibet. Yeah. So ectomycorrhizal symbiosis is around you and cradles you and holds you. We live in an area that is literally defined by this association. So pines, firs, oaks, tan oak, Sitka spruce, which is higher up but not so much around here. They're all the same. I mean, here, coast live oak, right? A bunch of different, there is tan oak down here. We have Doug fir down here. Those fundamentally depend on these fungi. But my question is, what shapes the community of fungi that are found in the forests, right? So there are so many different ectomycorrhizal fungi, but why are they found in specific areas? And what does that say? What does that association say? And this is kind of a growing, budding field of research that is investigating, like, what are the influences? Could it be soil? Is it biogeography? Is it the fact that these fungi are here, and so they're kind of isolated geographically from, say, something across a mountain range or across an ocean, and that's what's determining why the fungi are present where they are? Is it host specificity, the fact that some fungi are only able to partner with certain tree hosts because of evolution and the enzymes they exude that attracts each other and that allows for the formation of those root tips? Or is it soil nutrients? Is it climate? Is it the amount of water that a place receives? And honestly, across a lot of these studies, it's kind of a mixed bag. And that brings me to this map that I made this week in ArcGIS. I still got it. And what I did is I really just input the range of these 3 pines, because when I went to grad school, because I was lost. And I was like, yeah, this is going to be really good for me mentally, emotionally, and physically. And so I went to grad school and I realized that you can do a project in a different place and then have to pay for plane tickets. And that is expensive. And if you do a local project, especially in an area that you love deeply, like the far North California coast, you actually get to learn more about an area that you really love and do research in a place that is lacking in quantitative studies of this symbiosis. And so what I did, and what me and my advisor, Dr. Terry Hinkle, thought about was the fact that bishop pine, which is this guy, whose range is shown here in yellow, is pretty isolated at its far northern range from the nearest populations down south in Mendocino. And there's actually a pretty interesting overlap of ranges because shore pine, shown here in blue, is at its southern range limit. And so these 2 native tree species that have been co-evolving in Humboldt County for a long time meet in Humboldt and Mendocino County and a little bit in northern Sonoma County. And then we also have the case of Monterey pine, which is endangered in its native range and invasive everywhere else. Well, or tolerated everywhere else, but it's really invasive in Chile and New Zealand and places where it's been used as a industrial timber harvest plant. And so Monterey and Bishop are actually sister taxa phylogenetically, right? So they're really related. And what can happen with ectomycorrhizal fungi is if they come from an ancestor that has been associated with a certain tree host, usually phylogenetic proximity of host tree has an effect on the fungi that will associate with it. And so we have kind of a situation where I was like, okay, maybe I can quantify what's going on in these systems. Is the strongest influence the fact that there's a shared native range? Well, shore and bishop pine have the most overlap in fungal associates because they've been co-evolving in the same place. Will host specificity be the case? And will bishop pine in Humboldt County be most similar or more similar to bishop pine in Mendocino County, just inherently by being the same species? Or will the sister taxa Monterey, which co-occurs but is non-native in Humboldt County, will that be the most similar to Humboldt bishop pine? And so by setting up this study as so, my whole goal was just to be like, really it started with bishop pine because bishop pine is this like bougie species that lives in all the best areas in coastal California. And it's just kind of like, I could be a bishop pine, you know, if I was a tree, like that could be me. And so it kind of started from being like, okay, how does bishop pine in Humboldt County relate to everybody else? And what will it be most similar to? And how will that tell us about the influences on ectomycorrhizal communities? Does that make sense? Any question? Hey, interrupt me whenever. But these are pretty much my questions that drove this study. I just wanted to provide this slide as context, just to show you guys like who the players are. I think bishop pine is really beautiful in Humboldt County. It does occur predominantly on industrial timberlands. Shore pine literally grows on the beach. You'll be like on a dune ecosystem, and then there's like a hummock, and then it's just like deep, dank, dark forest full of fungi. Kind of an amazing ecosystem. And then there's Monterey pine, just on the side of the highway, which is fun. A pretty artificial environment. So these are my study sites. This is just kind of to give you an overview of how everything is spatially oriented in this study. So we have our bishop pine in green, Monterey pine along roadsides, shore pine here in a very fundamentally different environment than the compacted clay marine terraces that bishop pine grows on. And then my field sites down in Mendocino to quantify bishop pine were in beautiful state parks that I'm sure a lot of you guys who are a part of this club are familiar with. Okay, so in order to compare the fungal communities, you have to first quantify them. And so you have to basically measure who is like kind of like counting the fungi that are associated with the tree. And I wish that it was actually just collecting mushrooms, because that was the best part of this 2 years ago. But unfortunately, a lot of these forests, it's not just one pine or one species that predominates, right? So in my bishop pine field sites, I had bishop pine growing next to Doug fir, growing next to spruce. And so if I'm just collecting mushrooms and I'm like, this is with bishop pine, that's wrong. That's bad science. Yeah. And never mind. And so what I did is I collected mushrooms for the sole purpose of creating a voucher list of sequences with which I would compare my ectomycorrhizal root tips that I collected. So this was a fun process. This is Cass, who was helpful in sequencing the mushrooms. And basically, I was hooked up fat from the gel company. Greg Richardson, my father's company, who allowed me permission to use, just sent me, amplification and extraction material for mushrooms that I then used. I tested how successful my amplification was with gel electrophoresis, sent off to get Sanger sequenced. Anybody? Code on code? Yeah, Kat? Yeah. Yeah, actually, so fun. Just going through the little waves, being like, looks good, not clean, delete. Yeah, that was a good time. But really, what most of my data analysis is based off of is the presence of these guys specifically on pine roots. So I got really good at identifying roots. Pine roots have a reddish mantle with kind of this like peeling back, like dark brown, almost black epidermis layer. And if you scratch it, it really has this piney smell. So even in areas where there was like root overlap, became really comfortable with being like, okay, this is a pine root. And when I grabbed those pine roots and I took them to my lab, I was able to put them under a microscope with tweezers, pluck off the individual root tips, basically dry them out, and then morphotype them according to how they looked. So as you can see, ectomycorrhizal root tips are actually pretty morphologically distinct. And so all the ones that looked alike, I put in a pile. All the other ones, I put in another pile. And then I would take 2 to 5 representatives of each morphotype until I got a total bulk sample weight of 0.1 grams. Threw that in the negative 80-degree freezer until I had 48 samples from all of my 48 sample trees, 12 sample trees from each host. Humboldt bishop and Mendocino bishop being considered as different hosts. And then I sent them out with FedEx, and FedEx lost them for 24 hours. And then found them in Kentucky, but it was actually insane. And I didn't cry. I just disassociated and just kind of sailed through that period of my life. And it's just funny because I feel like that's very classic, very representative of a certain type of experience in grad school, where it's like, does it matter? Does anything really matter? Like all this work that you put in, and then FedEx is just like, but I will say that unfortunately, 8 out of 12 of my Monterey samples did not amplify, predominantly in this region. I'm not sure if it was sunny the day that they lost this box in Kentucky, but for some reason, those guys did not amplify super well. So I still have some data from Monterey. I have 4 out of the 12 samples that I sent. But when I'm going to talk about my analysis, my primary analysis on community composition is going to exclude Monterey. But I did do an exploratory analysis, which we'll talk about a little bit. But again, that's just how it goes. No worries. No worries at all. Oh, and then finally, this was a nice little color palette for artists in the room. I also had to collect soil from each place that I collected the root tips, because you can't just be like, oh, this community of fungi is associated with this tree just because it's the tree host. We know there's lots of studies out that organic matter, cation exchange capacity, or pH, nutrient content can also really affect the fungi that are present. So if I really want to say what are the influences on these communities, then I also need to quantify soil. Okay, that was all background. How are we doing? Good. Okay.
Carry on.
Carry on. Okay, so very cool. Lots of diversity in these ecosystems. Pines are obligate hosts. They literally cannot survive without their fungal counterparts. And so it was really cool to see that there's a ton of different fungi working together to make sure that these guys are okay. And there's also a beautiful array of morphologies of root tips. I did not Sanger sequence individual root tips, which is kind of a shame because I bulk sampled. I did next gen so that I could, actually get more data. But by doing this, I can't actually tell you what species of root tip this is. I can only tell you a bit a big list of all the species of fungi that were associated with each tree, which is fine. But we did find 101 different species from our 39 successfully amplified bulk samples. And then from 160 specimens of mushrooms collected, 76 were successfully sequenced. So that's not nothing. And basically we used those 76 to, for our sequences that we got from root tips, I blasted all of them against my mushroom sequences. Everything was blasted against GenBank, right? So it has an ID anyways, but by also blasting it against locally collected mushrooms from the same site, I also have pictures of the mushrooms for the root tip successful blast that I get. So it kind of it makes it a little bit more robust. Okay. Okay. Graphs, bar graphs. Easy enough. This is basically just showing you per host which fungi were most abundant. And something that I talked about last time that I thought was really interesting in these systems is that Tomatella sublilacina, this guy up here, is the most abundant. And it's this freaky little crusty racupinate crotacioid fungi that I still haven't found because honestly, it looks like a lot of other things that decompose wood. And I've taken a lot of pictures of things that look like this. And now those are just going to take storage up on my phone because I actually don't even know what it is. But that is the most predominant fungi in these ecosystems. And I think that's important to just pause and think about because what are the implications of that? When we have a fungi who fruits on the underside of usually decaying logs, can that inform management? Should we be keeping dead and down trees in a forest because the main fungal partner of these trees depends on that for fruiting and for sporulating? Maybe so, just to say. And then most common taxa across all sites, again, Tomatella at literally every single site. Different racupinate fungi, specifically Tylospora, Trichospora stellulata. And then some guys we know and love. Swillus umbenatus, slippery jack. Swillus tomentosus, also Swillus. I don't know if you call it slippery jack. Rusula, Cerulens, really common in the dunes. Different quartz. These are Dermosity quartz, so they can also be used for dyeing. DYEIMG. Terrible speller. And then, rhizopogon species. That's actually occidentalis, but that was the only picture of a rhizopogon I have. So just a sequestrate fungi that fruits below ground. Whoa! Wow. And this is, it gets crazier than this. So let's just start slow. Linear mixed models. Whoa, what is that? Yeah, multivariate analysis. It is basically I took 26 different soil measurements. I did it. Brookside Labs did in Ohio. And they gave me this data sheet back with, how much sulfur is here, how much copper is here, how much zinc is here. And it's whoa, this is great. I'm glad I paid $700 for that. But I actually need to limit the amount of variables I have in a multivariate model because it's distracting. Some of them could be collinear. Some of them could be kind of opposite each other and they could have different effects, which could skew the whole model. And so what I did is I basically did something that's really common. It's called AIC selection. And I did a collinearity screening. And I also used ecological rationale to come to a core 4 soil set, which is Bray P as available phosphorus, ammonium as available nitrogen, and percent clay as well as pH. And with these 4 variables, I basically ran a mixed model and then did partial regressions to find significant variables within this subset that affected alpha diversity. And when someone says alpha diversity, all they're talking about is species richness, evenness, and diversity. Things we know, things that we are familiar with. When someone says beta diversity, they're talking about overall community composition. Cool. So what I found is that in my significant partial regressions of these soil variables, predominantly species richness is what is affected. And species richness is really like, if you're saying observed species richness, how many species are found in a sample? And according to this and to pre-existing literature, I was really happy because this actually coincides with a lot of things that have been found already. When nitrogen and phosphorus are more available in the soil, there is less of an incentive for the host tree to partner with a wider diversity of fungi. And what will happen instead is the host tree will choose the more competitive, won't choose, but the more competitive generalist fungi that do well in environments where they don't need to be like niche farmers of nitrogen and phosphorus will dominate. And that seems to be the case in these systems. Evenness also, this one that has been highlighted in gray, evenness also decreased as phosphorus became more available in the soil. And so I was just interested to see this, and it made me feel a little better because I started doing data analysis. Chose not to take stats class. Yes, Katie.
I got a question.
Yeah.
So when you say the tree, the host, is more open to diversity,
Mm-hmm.
and chooses one, like how is that happening?
Yes.
Is the tree creating some sort of enzyme to like only accept something? How is that happening?
It's basically like the tree is desperate. Supply and demand. So demand is always high, or demand, like the tree needs those nutrients. But in these cases, supply is high. Supply is high, so the tree isn't like, I need you, Tricholoma, because you're really good at farming nitrogen.
I guess how is it rejecting?
It's not necessarily that the tree is rejecting, it's more that fungi, soil fungi or ectomycorrhizal fungi are also competitive with each other. And so these guys who are like, I'm not super competitive, but you need me because you need someone who's really good at farming nitrogen. Instead, the tree's like, I actually don't need you. And so it's not that the tree's saying, no, I'm rejecting you, but the tree is not like creating a space for that fungi to be like a serious competitor. Instead, it's being outcompete by, say, like Swillis or Russula or fungi that are better or more generalists at farming. We can talk. Okay, so this was my alpha diversity findings, but it wasn't really my main question, right? I'm kind of interested in community composition, how between these 3 hosts do these communities compare? Whoa, I told you it was going to get crazier. What is this? What is this? PCA, also known as, as a principal components analysis. So why did I collect those soil cores of just to quantify soil? Because, again, I can't just say that these communities are only due to the host tree. What I have to do is I have to account for soils. I have to say, when I am analyzing communities, I'm analyzing them in light and in regards to the soil structure at each place. And so instead of doing a linear mixed model where I'm just using a small subset of soils, a common thing to do is to ordinate the soils in a permutational space using a principal component analysis. And what this does is it does light collinearity screening, less strict kind of regulations because a PCA naturally does filter out soil variables that are conflicting. And so when we're looking at this beautiful plot, what we're seeing is how the soil environment of each host, this is Bishop, Mendocino, Bishop, and Shore. Remember, Monterey's out because we're just doing primary beta analysis. How they compare. And so what we can see is that Bishop, right? Bishop grows on compact marine terraces. Shore grows on sandy, beachy soils. And Mendocino Bishop actually kind of grows somewhere in between. A lot of Mendocino Bishop, especially McCarricker, is right on the ocean. And so what this ordination is showing us is how the soils relate to each other from each host. Does that kind of make sense? And what we can do is we can extract the axes of the PCA and input that into, also these variables are just saying like there's higher phosphorus, higher pH in shore pine, whereas there's more NO3N in the more compact soils of Bishop. But we can take these axes and we can put them in our distance-based redundancy analysis. This is a pretty simple looking graph, but one that I did cry over many times and stayed up. This is when I stayed up until 5 a.m. in my childhood bed. And this was a rough one because I have a really hierarchical study design, and so I wasn't getting enough. Basically, like I needed like enough variables or enough permutational power, and I had trees nested in sites nested by hosts, and so I was losing permutational power. And then I realized that I could do a differential centroid test, as one does. And pretty much what this showed significantly with a P of 0.0345 is that Bishop is more similar to shore pine than Bishop is to Mendocino Bishop. So that is kind of the main finding that is happening in these systems, is that a shared native range is a stronger indicator, at least in Pinus, right? We're just talking about in one genus, of fungal symbionts. And so we have, despite being in drastically different environments, despite when we saw the PCA, and we saw that those two soil environments of Bishop and Shore were pretty distinct, they will still share more fungal partners than Bishop populations will between isolated populations in Mendocino and Humboldt. And if we look at the phylogenetic tree of Pinus, it's subgenus Pinus, there's actually a ton of pines. Contorta really isn't that far away from muricata, muricata being Bishop, contorta being Shore. And so maybe it's not so surprising, right? Like maybe they're both pines, they're both in this subgenus, they're both in this section, Trifoliae. Maybe it does kind of make sense. But the fact is, they are like living in very significantly different soils. But that doesn't seem to deter the fact that they can share fungal symbionts and kind of act as reservoirs for each other in this opposite range, very small shared opposite range. Kind of cool. Right? And so then I also did an exploratory analysis with Monterey, just since I had some of those data. And what I found, I did a similar situation, right? This is familiar now. Set up a PCA, extracted the axes to account for soils in the redundancy analysis, and found that there were significant differences within this group. And when I did pairwise permanovas, and pairwise permanova is just doing one-on-one comparisons of communities, so compared an entire fungal community of Bishop pine to the entire fungal community of Monterey, and with each grouping, I found that Monterey was significantly different than all the other pines. And this surprised me a little bit because Monterey in Humboldt County is only 200 miles away from the northern tip of its native range. And in its native range, it actually overlaps with Bishop. And Monterey and Bishop have been known to associate with many of the same fungal partners, especially Suillus pungens. But Suillus pungens does not fruit, in my experience or in Terry's experience, with Bishop pine on the far north coast. And so even though it's really not that far away from its native range, the fact that, and this also accounts for soils, right? It's not just the fact that it's living in artificial environments on roadsides or as ornamental wind blocks. Its occurrence in Humboldt County is significantly associated with different fungal partners. And there's actually a lot of studies on Monterey pine, because like I mentioned earlier, Monterey pine is planted as an industrial timber product or species in other parts of the world. And there's a fair bit of research that shows that Monterey pine was not able to grow in Chile for a while. They were trying to plant it in Chile, and it wasn't able to associate with any fungal partners down there. And then they were like, oh, well, let's bring its fungal partners down. And then its fungal partners changed the way that soil, like microbial interactions occurred, and it started to detrimentally affect, especially Nosolothophagus, a relative of tan oak down in Chile, how that ecosystem basically worked. And so these invasions of trees and then bringing introduced ectomycorrhizal fungi is a very real thing. I don't necessarily think that's what's happening in this system, but I do think that maybe being planted whenever they were planted decades ago, they were planted with their own fungi that came from nursery stock or came from when they were seedlings, and they still associate better with those fungi than they do in this kind of, not really unfamiliar anyway, but this non-native range that they inhabit. And so I just thought that was kind of fun and interesting. And basically, what does it all mean? What does it all mean? Why did I do this? Let's reflect. Let's reflect on what we learned. Nitrogen, soil properties, kind of more of an indicator of alpha diversity or species richness. I've seen papers that have shown that there are community compositional shifts with shifts in pH. I didn't find that in this study system. That doesn't really, both things can be true. And really, my main question is that it seems like a shared native range is the most influential factor in controlling for which fungi are there. And then I think I talked about it more the last time I presented here, but I think it's really important to understand that these ecosystems are predominated by these really non-showy, just like crust-forming fungi. Fungi that we would otherwise think are wood decomposers are actually huge symbionts in these ecosystems and are like hugely important. And the reason why I think studying these things is important is because that basically native fungi are adapted to a range that they're in, and with climate change, ranges are proposed to shift, right? Ranges are shifting. Tree ranges are mostly going more north to deal with warming. And if regional partners from a native range are pretty influential in dictating who's going to be helping out those trees, as trees shift, Monterey was planted 75 plus years ago, but still significantly different than the native pines that grow in Humboldt. Will the symbionts of these trees be able to keep up? And there's studies, Kabir P., who's on my committee, that it doesn't look good. Like pinaceous forests in general are projected to decline, but the fungi that are associated with them are also projected to decline. And that can be due to climatic variables, but also I think because as things shift, we don't realize that things are so integrated and things are so symbiotic and things have been co-evolving together for so long that when drastic and rapid change happens, maybe they're not always able to adapt and to change with the speed of that change. I also believe that studies such as these are important for biodiversity and just accounting for what's in an ecosystem so that you can actually conserve it. And then finally, if you understand who the symbionts are of tree species, when you're wanting to do restoration or planting, you can use that knowledge basically to inform how to best help the success of trees. And so I tried to get through that pretty fast because I know we started late. And so that's kind of all that I'm going to talk about today. And I'm so happy you guys all came. And thank you so much to the Mycological Society of San Francisco and to other mycological societies that have helped do this research. And thank you to all my friends who have supported me, my family who supported me, because it's been so hard and it's almost done. And yeah, that's kind of it. Cool. Okay, that was rushed. But did you guys feel, are there any questions? Do you feel like you got it? Yeah. Yeah.
Were they using the next gen sequencing or was
Oh.
it also including Sanger?
Great question. Those are all next-gen sequences. The Sanger sequences I only used to create a database to blast the next-gen sequences against. And so when I'd get matches to the Sanger sequences, then I could literally be like, oh, this is MR87, Cortinarius tinctorum that I collected, and I have a picture with it. And so that Sanger sequencing of mushrooms was really just to add robustness to the data set. But yes, really good question. All the PCAs, all of this beta analysis and alpha diversity analysis is based on that next-gen bulk sequencing of root tips. Yeah. Thanks. Yeah, Kat. Hit me.
I'm curious about how much you know about like inter-year variation. So like if you know like of the mushrooms you saw above ground, how typical of a year was it? And like do you think the tips reflect that?
Totally. Yes. And I only collected tips. I'm writing my discussion right now. Saying one of those things is like I collected tips from July to October. So that's a fourth to a third of a year. And there's totally turnover. Like if this could have been it's a 3-year study, but if it could have been a intentional 3-year study or maybe, longer, where I could have collected root tips at different times of the year, even at I collected them all at the same horizon, but if I could have done a differential testing of a more organic layer horizon versus a more mineral soil layer horizon, I think that could have changed. I think that could have changed it. I think the fact that there are significant differences in the community, just looking at it from this little glimpse, maybe does indicate that maybe that would exist in other times of the year, or maybe it would exist if I had a fuller data set of a whole yearly thing.
Well,
yeah.
It's still awesome. Don't get me wrong.
Yeah. No.
No,
I know. And then I was reading papers and I was like, oh yeah, being really intentional about really taking root tips from different places. Because a lot of the studies on root tips, it's this was a 5-year study and we went back 2 years later and we collected more root tips. And because even community turnover within 2 years is a possible thing, especially based on drought or like that. Yeah. A great point. Yes.
This is a really rudimentary question, not in the society, and then ever hearing about this. How long do the mushrooms have to be in relationship with the tree for the tree to have been?
That's great. I mean, Kat, maybe you can speak to that.
I don't know if that's
Yeah, but they're long relationships. These networks, these soil networks that exist between the mycelium of the fungi and the roots of the trees and connecting different trees with other trees and other fungi. They can be really robust. They can last for a really long time, and they can be super established relationships where I would go into a shore pine, a shore pine setting, into that dankety dark forest, and it would just be OMG, there are so many of one mushroom just in this area. The mycelium is everywhere. These guys have been rocking and rolling for who knows how long.
And even if you pluck the mushroom out, the roots are still there, doing the work. So it's not taking anything away from this relationship if you pluck them out.
You bring up a good point. I Where I live in Humboldt, it's like forage, and it's a lot of it's like there's a lot of people who forage to sell. And so there's a lot of people who will pick the patch, if you will. And actually a great amount of energy, especially in the form of carbon from the tree host, goes into fruiting. And fruiting isn't just to be, fruiting happens so that sporulation can occur. And sporulation occurs so that new genetic individuals that might be better at adapting to a warmer environment can then grow. Because when these guys are growing from their mycelium, even though they are in a dikaryotic state, meaning that two nuclei are not fused, allowing them to be a little bit more flexible, it's still like one genetic individual. And so it doesn't hurt to take the mushrooms, but it is good to leave more mushrooms than you take because they're sporulating and they're doing something important. It took so much energy to create that mushroom. Yeah.
On that topic, so about 20 years ago, I found this really good porcini patch here in the city with Bishop Pine.
Mm-hmm.
And I picked it really good and heavy, like 3, 4, 5 years.
Uh-huh.
Now there's none.
Well, they
I never left any. I wasn't the only one picking it. Other people didn't spot it.
But that's I mean, ain't that the way? Ain't that the way? Just extract. I mean, not you, sorry. Because it seems like that really.
They are picking too.
Okay.
Once.
Yeah.
I actually ran into JR at the spot.
Oh, the spot's blown. Susie.
So one thing there's research that there's actually succession from the younger root tips inward to the tree. So there's a different suite of mushrooms that show up close to the tree as the tree ages than we see out at the edges of the tree. So there apparently is some differences in ages of the mycelium.
Totally.
Depending upon, within that are associated with an individual tree, depending upon where it's collected.
The same thing is true with seedlings. Seedlings of a tree will usually have a pretty different composition of fungi than more mature individuals,
Right.
Which kind of goes into what Kat was saying about like, where are you sampling? Because when I was sampling for root tips, I had a standardized protocol for sampling, and I sampled 6 inches from the base of a tree. And so I'm sampling, and granted, like young roots can still be near a tree. But it's
Right.
just to say to your point that I'm glad that I did find significant differences because maybe it indicates that even on that scale, you can
Right.
detect it.
Could be a variable.
Yeah. Susie.
If you could visit these sites again and do the analysis of these same sites, the same species, you're gathering again in another point in history, when would you want to choose?
Like back in time?
One or sort of.
Yeah.
So you said
Yeah. Or are you talking about like, can I say 1850?
Yeah.
Okay. Bishop Pine, old town foresters, basically have documented, old time, like early 1800s, Bishop Pine that was 6 feet DDH. That means diameter at breast height. That's right here, 6 feet around Bishop Pine. And so to sample old growth trees, because also some ectomycorrhizal fungi will only grow in old growth ecosystems. And I'm sure like what we've been talking about turnover on mature roots, on less mature roots, seasonality. I wonder if like in older ecosystems that have had more time to evolve, if you would just see like this crazy, just more diversified, richer fungal community. And it brings a tear to my eye to think about what has been done, because CDH, I've worked a lot of jobs, and I believe in the fact that we are standing in a wood building, but it is sort of like, yeah, we're just going to clear cut this guy, and it really changes things. And I know that that's a management tool, and I've gotten in arguments recently about it and I don't know. It just seems like if you're managing something as a resource and not really thinking about the ecosystem, you will inherently not really be looking out for its bestness.
On a lighter note, so for several years now, we've been going to our friend's private property down on the peninsula, and the chanterelles are prolific.
Nice.
And the rule is we only pick half.
There you go.
Yeah.
There you go. Wow. You can, Teresa.
Yeah, when you started, there was the idea that I got anyway, about the phosphorus and the nitrogen having driven the generalists and the best farmers.
Mm-hmm.
And I wonder how much the soil is changing.
Yeah,
I
with
mean-
management or just in general? With time.
In general, and how it would affect that, change the relationship again, because all of a sudden,
Yeah. Well, a lot of actually experimental studies, and actually a lot out of China, nitrogen, fertilizing the forest, fertilizing the forest, fertilizing the forest. That's just adding nitrogen. And it was like studies conducted over 20 years of like intermittent fertilizer addition, and where they did indeed see a decrease in fungi, or at least in species richness of fungi. And so how are these environments changing over time? I mean, I don't know if anybody is putting down fertilizer in these forests. That'd be weird. But maybe that's an idea that someone would have. And now you can say, no. A talking circle, you can say, no fertilizer. Yeah. Anybody else?
In your wonderful ArcGIS map,
Let's go.
there's a population of bishop pine on Monterey Peninsula
Yeah.
and close to a native Monterey pine
Mm-hmm.
forest.
Mm-hmm.
So would you expect to find those bishop pines having a similar suite of fungi, more similar to those Monterey pines than to the bishop pines further north?
Definitely. Kat, who's in Bruns's lab, studied bishop pine mostly up here in North Bay, but they would find them, and another researcher found that they did share a lot, especially Swillis, like Swillis pungens, a lot of rhizopogon species. Like their communities are strongly overlapping, and they have the added element of being like more phylogenetically closely related. So yeah, I would think that they would be really, really similar. And I think it would be interesting to even quantify, like compare similarities between two overlapping populations to be like, well, these ones are more phylogenetically proximate. Like, will they be even similar?
Yeah.
But I'd like to get paid to do it. I'd like so much payment to do that.
Fingers crossed.
Yeah, a lot of money in the world right now. A lot of money being printed. Oh, anybody? No, nobody. Okay. And thanks everybody for your patience and for staying. And it is 9. It is 9 o'clock. That's okay.
Thank you, Monica. Great presentation.
know what that is. What if that kind of mushroom get introduced into the environment like that?